and we are coming down to a depth. This waypoint is at 530 meters, the one that we're heading to.
We're actually at 725, I believe.
Yeah, so we're getting close to it, as you can see. But in this area, we have a very swiftly flowing, significant current, long-term persistence. It has changed over the course of geologic history. It's changed its location. It's changed its strength. Right now, we know it's very strong. We're being affected by it. So that strong current is likely preventing those very fine particles from settling. They're just sticking in that current and traveling along with it. They're still suspended. So what we will likely see is a coarser grained material, and the grains are probably going to be biogenic in origin. They're probably going to be mostly the hard part remains of organisms that lived in the surface waters. Yeah, we're a little far from shore to receive any of the, well, we'll receive some maybe, but the material that you think of on beaches, the sands that are made of quartz and other mineral matter, as opposed to the microfossils of organisms that once lived. So we'll try to zoom into this sediment once we touch down, or different points along the way, to see if we can see some of these larger microfossils.
And one thing that the corals, the Scleractinian corals, colonial corals that build a framework, they baffle some of those particles and actually knock them out of the water column, which helps build these mounds over time.
Go ahead, RV now.
Just wanted to let you know that the vehicles are on the bottom, and I would like to confirm that the center rotation is set to the A-frame, please.
I will shift the center rotation to A-frame now.
Copy that. Thank you, Bridge.
Center rotation is now A-frame. Do we have a bailout?
Yes. We would like to head dead south as a bailout plan, if that works with you.
Yeah, bailout is set to south.
Either south or forward ahead, anywhere between south and southeast will be fine.
Sounds great.
Thank you.
So it does look like we have some coral. We have a little bit of dead coral structure we see right below us and a little bit to the left. And it looks like we're looking at quite a steep slope here. And we're going to let the...
What is that fluttering down there?
Looks like we might have a sponge or two and maybe a little fish in the center. Again, we have to let the ROV kind of settle out here and get their bearings before we can start exploring. But this is an exciting start.
All right, pilot, navigator.
How's it going,
and our pilots informed us that we have about a knot of current down here on the bottom, a knot to a knot and a half, where the surface was more like 2 knots. And so one of the games we have to play this whole dive today is the ship is getting pulled by a stronger current than the ROV on the bottom. And so we're going to have to balance those forces and movements. And we have a sponge here, white demo sponge most likely on the left. And you can see basically old fragments of coral are the darker things that you see all over the seafloor here.
1 sec video, we're still
and that's typical for the mounds in this area. The bases of the mounds are eroded coral and older coral that has fallen down from the top of the mound where most of the live coral is. and it's probably growing best up there because of the swifter currents and being so elevated in the water column. The two little red dots you see in the middle of your screen are lasers, and they are 10 centimeters apart, and we use those for sizing. So even though we have those for measuring, we seem to still be surprised about how tiny the collections we make are when they come to the surface compared to what they look like on our video monitors here, because we have such great zooming ability and everything looks more grand in general.
Hey, Nav. So Nav, I'm going to back up a little bit in the co-pilot because I think the layback keeps pulling him away from me.
It does. Especially, it's looking like you don't have very much on bottom.
Oh, there's
here.
a nice big sponge and another
so I suspect now that you guys
part
are
of
acting
dead
as
coral and
kite,
maybe one live little branch or two.
That decrease.
Copy that.
Oh, and a chimera.
Ooh. Hello.
Video,
wow.
You want to go
Morning.
Absolutely.
That sure is a great thing to see at first.
Nice to see.
Pretty.
Wow, beautiful.
Nice.
It looks kind of small. Is that kind of a juvenile? How beautiful. Oh, that's a great shot of all the dead coral behind him, too.
Is that kind of coloration normal?
I think the coloration pattern looks different than what I'm used to seeing. And again, this one is quite small compared to what I've seen in the past. Also, this appears to be a juvenile.
Hey, Copa, so what I think is happening right now is layback is pushing you back, so away from me.
I don't know if we have
so
any
just keep an eye out.
Fish experts on the line or in the chat room that could tell us, confirm that.
Yeah. I backed up a little bit, but...
We also have some octocorals right behind him.
Back 200 meters.
Or her.
Yeah.
Yeah, once you're done with this, Sean, go ahead, come back in towards Sirius.
Center on the front of the face.
Yeah.
You can see how he is.
Video is clear.
Most of the time he's been facing toward the camera because he's facing into the current. And you can see from these octocorals in the foreground that they are being swept with the current. We have the current behind us. We're facing almost due north at this point.
He really seems to be interested in you.
Yeah.
Wow, and that's a gorgeous scene he's swimming through, too. Trying to see what kind of coral that is. Wow, look at that. The big stands of mostly dead coral.
All right, Washleads.
Bright yellow sponges.
On bottom, as you can see, I'm just centering up with Sirius as we settle. But after that, we should be good to start the dive.
Sponges.
Yeah, the sponges seem to be doing well here, but not as many coral. But the coral are still holding on to some of these dead bundles of coral.
And we're quite far away from the corals, but to me that looks more like Analopsamia profunda than Lophelia pertusa, but we'll have to get a closer look to confirm that.
Come back down. Yeah, it works. Yeah, you can, if you feel better keeping it up, go for that.
You can see that there's this whole mound is covered by little fragments of dead coral. All of the pieces you see on the seafloor.
All right, watch leads. If you see anything, just
lots of
let me
nice
know.
sponges. And the sponges are also filter feeders, so they're looking for a similar environment that the corals are, as far as being elevated off the seafloor and trying to get as much real estate into the water column to capture food that's passing by.
Oh, you're talking about high pack. Well,
no, relative D2. You can feel
kind of east.
Well, now it's going kind of that way.
East to west, huh?
Yeah. It's a decent amount, though. You do have to fight it.
Well, this is great to already see so much on the seafloor. You can really see the topography here. Yeah. What we're trying to accomplish here, if you're just joining us, is to characterize these mounds that occur throughout a huge region of this part of the Blake Plateau off of the Florida-Georgia coast, referred to as the Stetson Mesa.
Yeah, we're generally heading north.
They're like
put the first one at
watch one.
Mounds,
go ahead.
As well as strings of mounds that sort of coalesce and undulate along the path of the current. And we were hoping to find just this. You can see the mound topography.
Co-pilot, do you have a bearing towards
the biologists?
Well, I'll just send you a little bit.
How far up? How far up?
We'd find living corals and sponges on top of dead remains of these organisms.
Yeah, we could do the one in the middle. We're not quite far enough to get to the top.
Entire mound may be a very slow accumulation of these organisms and their remains, basically.
So it looks like we have, an octocoral here.
I like to listen.
A little bit.
All right, video, go ahead.
A little bit to the top of the screen, and a cutthroat eel coming in. And here's our first good close-up of the coral.
Is this what you wanted,
Yeah.
Ashley? Great.
Bridge, ROV nav.
Let's see.
Go ahead, RV nav.
Yes, can I please get a ship move?
Ship move,
30 meters, bearing 315 degrees, 0
this
decimal 2.
does look like an alapsamia to me.
That is 30 meters, bearing 315, speed decimal 2.
Good copy.
But actually, the coral to the left in the back looks more like madrepora.
You're starting to pull me.
Looks like there are multiple species here of the white stony coral. Then we have an octocoral behind that.
Copy, bridge.
All right, video, come wide. Thanks. Just gonna go off stick for a second.
Both sonars set to 30.
Great.
I don't know if the octocoral looked like a bamboo. Didn't get quite a good look, but we have lots of things to look at, so I'm sure we'll see more.
All right, watch these. I just need to back up for a second to align myself better with Sirius, and we'll be back on the ground in a second.
We're at the top of one of 3 mounds that we hope to cover during this dive. We had intended to begin at the base of the mound, but the Gulf Stream was already changing our plans. But now that we're down here, we should probably be able to
back a little more,
complete the rest of the dive without too much havoc.
Going back down?
We'll see.
Yeah.
Fingers crossed.
But even this view is great as we fly along, because you can see the extent of the live and dead coral, and the cover of this whole mound is at least dead coral. So again, it kind of points to a biogenic origin of this.
Definitely.
All right, watch these. I'm back down on the ground. Just let me shout out if you want any zooms.
On the chat room, we have some of our geologists, Jason Shader from the USGS, Scott Harris from the College of Charleston, and Amanda Demopoulos, also from the USGS, and they're curious about the sub-bottom profile. When we run our transits to and from sites and sometimes across the sites, we also use a sonar type of instrument that can penetrate beneath the surface sediments and give us an idea of the character beneath the seabed to see if there are layers of rock or if it's a biogenic mound, and a fuzzy return tends to at the surface tends to tell us that it is not well consolidated, and that these might be
partial.
These might be biogenic in origin. I'm not an expert on this. We're hoping to find the records from the 1402 Okeanos Explorer expedition that had one of those, and I know we have them on board. I just don't have them on hand, and we'll try to get them to our geologists online so they can give us a great interpretation of them. These mounds definitely have a very different character as far as that sub-bottom profiling indicates, that the areas between the mounds are flat-lying and have different sediment layers, which is likely characteristic of most of this very broad, expansive Blake Plateau area.
And now that we get a good look at this coral, and you can see the way it is formed, it does look like Lophelia here. Very, very thick skeleton Lophelia. But that white coral in the middle is a Scleractinian colonial coral, Lophelia pertusa, most likely.
And that's one of the stony corals that you find in abundance on the shallower edges of the continental margin. Is that right? Yeah.
And
I know
especially
we've seen it north of here and shallower.
Mm-hmm. It's quite abundant from here up to about North Carolina, and then we had a we had a gap in its known distribution in the Mid-Atlantic region until our Mid-Atlantic Canyons work a couple of years ago, and we found it in the canyons, but in a very different growth form. It was actually growing, hanging down from ledges, and so it was very hard to find until you could go and look under ledges with something like an ROV. You weren't going to find it by just dropping a camera or trawling.
You might want to try a little more mowing of the lawn back and forth along this ridge, and that way you can kind of cheat a little bit. And
I think a lot of people who might be watching are
on bottom, that way all that stuff's
maybe new to the idea of this deep sea coral. You think of corals as something that must live in a shallow environment with lots of light, tropical conditions, and here we are on the seafloor where there's no light.
Go ahead, Arvina. I was actually
definitely not tropical temperatures.
I was going to say, it sounded like you clicked in there.
I didn't want to interrupt you. Position complete.
Roger, thank you.
Quite cold, definitely not tropical. Strong current flow, and these are what are referred to as deep sea corals. So Cheryl, can you just give a quick overview of what a deep sea coral is and the definitions that you all use?
So a deep sea coral is any coral that lives below 200 meters. So it's below the photic zone in areas where there's no light, cold, stably cold and dark places. And so the tropical corals have zooxanthellae, which photosynthesize and create food for the coral.
Algae, microscopic algae.
Right, microscopic algae. And of course, those aren't going to help the corals that live in the dark and the cold down here in the deep sea. So they do not have those, and instead they rely solely on capturing food particles from the water column.
Copy. I'm not going to come back
so
anymore.
It's amazing the diversity of corals that are down here in the deep sea. Over half of the known species of corals actually live below 200 meters. And of course, they're a bit more of a mystery because we're still trying to identify all of them that live down here. We especially know less about the octocorals. They're very diverse, and those are the fan-type corals, the gorgonians, and we've been identifying species of gorgonians at a faster rate than we have been of the scleractinian corals in the last 10 years or so. But we're still finding new scleractinian corals also, and a lot of the diversity of these stony corals, scleractinians, are in the solitary cup coral form, and so keep your eyes peeled for little bitty corals on the seafloor or attached to other things. And those might be cup corals that are that add a lot of the diversity that we see.
Can we repeat that
You can
move,
see
please?
a Sideris pencil urchin in the upper right.
That's a good copy.
Sorry, we have a request if we could zoom in on the non-scleractinian corals when we get another chance, just so when you see one, if we can zoom in.
Yeah, just let me know. Watch it.
Right next to the sponge.
Yeah, sorry. Video 1 sec.
Yeah, because they are fewer than the others.
1 sec video, let me try to spin.
There's a great pencil urchin off to the
Thank you, Bridge.
upper right of center, too. We might want to take a look at that while we're here.
Mm-hmm.
We can look at this sponge, too. Nice view of the horizon. You can see these organisms reaching up into the current.
Mm-hmm.
Nice job.
And Eric Cordes from Temple University pointed out that the coral frameworks are very good at scattering sound, and so they've gotten some low reflectivity returns on coral mounds with seismic data in the Gulf of Mexico.
Permission to zoom in on Sirius?
Yep, and that is a little octocoral.
Permission to zoom in on Sirius?
You can see the 8 pinnate tentacles on each polyp, and that's the pink organism below the sponge.
So, based on those little things, I'd say current.
We also have some hydroids in the upper right.
Look at some of these particles. So it's it's a little more east-west down here than north-south. All right, good to know.
And this might be an Alcyanacea. Duva florida. Duva would be the genus, and Florida would be the species, is what Sandra Brook has commented.
Shot including the sponge.
Copy that.
It's a beautiful shot from Sirius.
Oh, and if anybody is switching between the streams, Stream 2 has a nice view of D2 from the Sirius, looking at this nice coral in front of us. So there's an amphipod. Somebody pointed out.
All right, you could come wide.
There's an amphipod on the octocoral. Jill Bork from the USGS. That was a great shot.
Currently back 175 meters.
Copy 175.
It's decreasing.
Yeah, this current allows us to touch down, but only briefly before we have to move on.
And Chuck Messing noticed that there was a tiny feather star crinoid inside the sponge that we were previously looking at. I didn't quite see that, but So as we explore the top of this mound feature, we see more of the dark stained dead coral structure with little tips of white coral, which is the part that's still alive.
Once you do that, pilot, confirm that's all set, then I'll zoom past the skids if that's okay.
And then probably check the rock boxes as well. Send them outboard.
Yeah, video, I copied that. All right, thanks.
Pushing past the skids.
Yep.
A little more tilt up, we can get a wider shot without the skids too.
Is that good for you?
It's improved, yes.
Go ahead, Watchman. Yeah, dead just coming into view at the bottom. Is that what you want? Great. All right, video, give me 1 sec.
So it looks like we might have some more of that Duva, Florida here.
All right. Video, you can go for zoom.
See the dust coming in from the left?
Yeah. It's just going to go get blown to the west.
And we have a couple of sponges.
Copilot, you can kill laser, or never mind. Zoom past them.
Yeah, I'm trying to get that clump on the upper left.
One upper left clump.
And as you notice, the more you look around and zoom in, the more organisms you see. The dead coral structure creates an amazing number of nooks and crannies for other organisms to attach and live. So even after the scleractinian coral is long dead, it's still creating habitat for other organisms. Looks like a tiny brittle starfish on the upper left of this soft coral.
I don't think we're close enough, but can we try those extra lights?
The lidar? The fog lights.
The fog lights, you guys.
Yet another look at the coral skeleton that this is growing on. It might be an Alapsamia.
Yeah.
As I said before, both Analapsamia and Lophelia are common in this area. As we move further north, the Analapsamia will become less common.
Much nicer.
But here we could have a good mix of both of those corals.
Copy.
Going for full screen.
Yep.
Tilt down.
Tilting down.
And Tina is asking if it's not Dendrophilia. I'm not sure which coral she's referring to. Is that the soft coral?
And the video's clear.
All right, go ahead.
Is there a pycnogonid at the bottom of that white sponge? Right above at the top screen.
Oh, and there's a little crinoid at the bottom that we just saw.
You see it? That's centered now.
Yeah, that might be a little bitty pycnogonid on the sponge right in the center of the view right now. I don't think I've ever seen a pycnogonid that tiny.
Video's clear. Video's clear.
Copy. All right, video can come on. Killed fog lights. Copilot, thank you.
ROV 9 bridge move complete.
Thank you, bridge.
We have another cutthroat eel, synaphobranchid, in the center there. And several more coral structures ahead and sponges. And we have a yellow coral on the left here. Now, in my experience, Analipsamia in this region is often more yellow. So maybe that's Analipsamia that we're passing there on the left. But Analipsamia can also be white, so it's kind of tough to tell the difference between the Lophelia and the Analipsamia when we don't get a real close-up view.
Copy that. Copy. All right, video, go for zoom.
So we're taking a look at this guy. I don't know if this is the same individual, but
iridescent almost.
Yeah,
beautiful coloration.
I haven't seen this coloration, and also this is much smaller than any chimera I've ever seen. I don't know if there are any fish folks out here that could confirm that this is a juvenile. And this looks like a different one than we saw when we first sat down. Yeah, very different coloration.
Max zoom.
Copy.
Very nice, stable shot.
Yeah, that's because I'm set. Can't take credit for that. Go ahead, wash one.
You copy that.
Right,
and again, you can see that the seafloor here is sediments of light color, but also all these coarse, rubbly pieces of dead coral. And
full wide.
Very likely this mound is made of just long-term piling upward of the dead coral pieces.
And that's exactly what we were hoping to confirm.
Exactly, yes. Because this is one of hundreds and hundreds of these mounds along this mesa, and this is the first of 3 we hope to traverse today, and we'll see if all 3 look similar.
Anything specific
here?
If they
do, it gives us a really good indication of what this whole area may hold in terms of the biological communities.
Is it white? Copy. Yeah, give me a second.
Zoom in on some of these organisms to give you a close-up. Again, the laser beams you see on the screen are 10 centimeters apart. Give you somewhat of a sense of scale, even though it's really hard to fathom how, oh, no pun intended. It's really hard to imagine how large or small these organisms are.
But one thing that it would be easy to imagine is if a trawl net were dragged across this surface right here, both the sponges and the corals would likely be swept off, and both of these types of organisms are very long-lived. It might take decades for them to come back if they do at all. So that's why these habitats are so sensitive to things like disturbance from trawling.
Video, go for zoom.
A little bit left.
Copy.
So this is an octocoral. And I wonder if Rachel Bassett might be able to help us with an ID on this one. She wanted to take a closer look.
Copy that video.
And it sounds like we have the group that Rachel's working with now kind of stumped about what this octocoral is, so I'm not the only one. And while we're looking at that, we also see little kind of tree-like organisms that are blowing around in the current. And Chuck Messing was noticing that some of these are dense populations of agglutinating forams, foraminiferans, Astrorhiziacea, that are covering the dead coral branches.
Give a little push forward.
And Chuck has hypothesized that they might actually help accelerate the sediment deposition on these mounds.
Copy.
So they collect the fine particles that normally drift through the coral branches, and then they take them in and hold on and take more sediments out of the water column.
Yes, there are many, many species of foraminifera, these single-celled organisms that live on the seafloor, the benthic foraminifera.
It's like a different branch one.
There are also many that live in the sea surface called planktonic foraminifera, and their shells are settling down here as well.
Yeah,
right there.
Dead center. Yeah, let me rotate around.
Do we want to zoom in to one of these?
Copy. Yep, you got it. Just give me one sec.
ROV nav bridge.
And we'll hopefully see some more of these agglutinated benthic forams. They're very unusual, well, not unusual, but they're in a different group of the type that we see in the microfossil assemblages.
Okay.
They are almost arboretus. They grow in fronds and attach to their chambers whatever sediment is available.
Hitting my toe on its neighboring.
And we have another Synaphobranchid cutthroat eel swimming by.
All right, video, you want to try that?
Here's another octocoral.
Awesome, thank you.
With hydroid in the background. And this looks like a bamboo coral. You can see those brown bands that alternate between the white calcareous parts of the skeleton.
Maybe Scott France can identify the species for us.
Fog lights.
Is he online still?
Sorry, video. I kind of drifted a little bit. Coming back.
And Chuck Messing said that perhaps this might be a Unicella, which there's a similar species on the Miami Terrace just south of us. South of us quite a ways, actually, but. And the pink organism in the back left might be a cup coral with its tentacles retracted.
Any idea what the purplish pink thing is to the lower right? Just tilt down a little bit and slightly right.
Yeah, I'm not quite sure. There's a
at the bottom of
purple pinkish
right corner of the
organism to the lower right, and
right there.
It might be something like an anthomastus that's retracted, but
it's pretty steady around 190 now.
Okay, the Unicella might have been the white octocoral that we looked at previously, not this one. Thanks, Tina.
Back soon.
Copy.
Look at that little puff in the upper right corner.
Upper right. Copy that. Yeah.
Or maybe that's one of the Duva Florida that's also just contracted its tentacles. That might be what that is. So much life to look at here. It's rather amazing. We have hydroids. And
yeah, you can see the particles getting the manips right now.
Okay, pulling it out for a full image of this area.
Copy that.
You can see how the
tilt.
Remains of the dead coral have turned brown, and that brown is the iron oxides, maybe iron manganese oxides that are in the seawater as ions,
you have the
and
ability
they precipitate
to rotate that
onto
bright
surfaces.
Object to left of center.
So a freshly dead coral or a living stony coral would be bright white, and as it has recently died, it might be very light brown.
At the top?
Similar to what you're seeing.
Okay, I was going for the wrong one.
The foreground, and then as it sits on the seafloor for long periods of time, it turns a dark brown, almost black. So you sort of see the stages of fossilization. Looks like there's a brittle star also winding around the base of this. At least one, maybe two.
Yeah, I see several.
You see the arms.
And it does look like that is a cup coral of some type. They are quite difficult to distinguish to the species level without collecting them and looking at the morphology of the calyx, which is where the polyp sits. And just got a flash of a fish. There he is.
Beautiful.
It's a cutthroat eel. And also in the
Very
lower left are two different types of sponges. It looks like a demo sponge and a glass sponge, which is the more clear one. And then you can see several other sponge types on the horizon. And there were some small feather stars in that group that we were looking at. Again, this dead coral structure just creates habitat for so many species, and the more we zoom in and look around, the more you can actually see. Oh, well, that's great. Nice big group of live coral, this white coral ahead here.
Co-pilot, ready for move? All right, this one's going to be 320.
Go ahead, watch it.
Bridge, RV nav.
Go ahead, RV nav.
Yeah. Another ship move, please.
Okay.
30 meters, bearing 320 degrees at 0.2.
Video, let me
30 meters.
stand by.
320, speed decimal 2.
I think I'm going to have another cutthroat eel. Trying to take a closer look at this coral. We're getting pulled a bit, though.
It's just me turning to try to get the dust to blow away from it. Watch it.
Okay, great.
It's interesting when you see coral reef communities in the tropical shallow regions, and you see white coral. That's a bad sign.
Right.
Thank you, Bridge.
It means that they're symbionts, the microscopic algae, zooxanthellae, that live among their tentacles.
All right.
It means that they have vacated the premises. Corals don't do well without them. So that's referred to as coral bleaching. But in these deep sea corals, bright white means healthy.
Yep.
And something to get used to at first.
Video, depending what he wants to play.
You can see how healthy those are, and the darker the coral, certainly that crusting of the iron oxides means that the coral polyps no longer live there.
Yeah, and again, this looks like an Alepsamia profunda, and you see the dead matrix is the brownish part of the coral skeleton to the left, and then the white part with the polyps is the live coral.
Okay, pulling for full screen.
Copy.
It's a great shot. It looks like a hydrozoan growing off of the top of that coral. And I wonder if the bottom right, it looks like that might be a different coral.
Tilt it down and slightly right.
Is it on screen?
I think that's what they were talking about.
Copy. Is it on screen?
I think that's what they were talking about.
Copy.
ROV now, bridge, move initiated.
Thank you, bridge.
Copy, let me know. I'm just going to go for this coral. If I tug you, just let me know.
Wonder if that's actually a sponge instead of a coral. We will soon find out. And is that a bottle on the lower left? The first marine debris that we've seen for the day?
We didn't see any yesterday, I don't think. Or 2 days ago at the
we might have seen one piece of paper or something, but that was it. Yeah, that looks like a sponge.
Awesome.
Beautiful color. I hope that Shirley Pomponi will join us today, our local sponge expert. It looks like more hydroids behind this sponge.
Okay, go for full screen.
Copy.
Another cutthroat eel, and you can see the dead skeleton of an Alipsamia profunda there on the lower left, and then at least 3 different sponge species it looks like, and then maybe on the right side was more of that Duvia florida, soft coral. And we've kind of reached a flat portion of this mound, flatter than we had been climbing anyway.
The fish in front of us looks different. It's got a black tail.
Yeah, if we could zoom on the fish. I'm not sure if that's different or not. It looks quite similar to a Synaphobranchid, but it could be a different species. He's swimming right towards us, right through the coral. Oh, and look at that nice big hydroid that he's swimming past there. Big feathery looking hydroid. Again, it looks like there's some little white dots on the center of that, like we saw previously. Oh, and we have another one of the white octocorals in the lower right. After we check out this eel, maybe we can look at that.
Tilt up.
They're well adapted for poking their nose into this type of coral, aren't they?
Yeah.
Just let it swim through the screen.
Copy.
Yep, it's going to float away. He's floating out of the image.
Can we get the lasers just briefly so we can get a sense of how large the eel is? I think he's probably about 10 centimeters, but we'll find out. Oh, he's longer.
He might. Ooh, here comes a friend.
Yeah, so
I do think that's
about 30 centimeters in length.
A Synaphobranchid.
30 to 40. Thank you.
Welcome. And then, you wanted a view on something else in view?
Yeah, there was a white coral just straight ahead of the eel. It's an octocoral, a soft coral, and it's white.
Copy.
Immediately right of your lasers, I believe.
Is it?
Correct, just to the right of where the lasers just were. Yeah, right there.
Copy. Stand by. Video. Give me a sec.
I wonder if this is what we were looking at before, a little further down the mound. And Sandra Brook commented that these Synaphobranchids are common in both Lophelia and Analipsamia coral habitats, so this is exactly where we would expect to find them.
Zoom from here. And Copilot kill lasers.
Yeah, there we go. Yeah. That looks like the species we saw previously, which I think Chuck Messing thought was Unicella, maybe?
ROV now, bridge move complete.
But I think there was quite a bit of debate.
Bridge.
I don't know if there'd be any interest in taking a small sample of this.
That looks so different with the skeletal covering being so much more opaque than the tentacles.
Yeah, this species does have a lot of tissue over the skeleton, kind of thicker tissue than some of the octocorals that we've seen.
Going full screen.
Copy.
So Rachel has commented that a sample of this would be a good addition to our collections. Pilot, would that be something that would be possible?
Give me 1 second. Watch lead before I answer. Video come on.
Again, one of the things we have to balance here in the high current is the stability of the vehicle and the position of the ship. So we'll have to see with every collection whether it's going to be feasible or not.
Unless I get it.
I think you can do it. Get up lateral a little to your right.
And Chris Kelly has joined us, and he agrees that it's Unicella.
Come at it from this angle?
Yeah, if you... I'd hold that angle and just lateral to starboard a little, and your toes should get around those two big chunks.
Lateral starboard, you think?
Starboard.
Sorry, stand by.
Right. Just a foot or two, and then your left toe will be clear of that chunk, and the right one should be clear of that big grouping. Yeah, right about there-ish.
Alright, stand by.
Yeah, right there. I think you got it.
Alright, Copilot, can I borrow your hand and hit joy lock? Thank you, sir.
You can also see a little pink organism just back.
All right. Yeah, Washlead, I think we can grab that sample if you'd like.
Great, roger that. Thank you. So we are going to attempt a sample collection here of this Unicella, the octocoral that you see kind of left of center of the screen here.
Copilot. Is what's ship status?
Yes, and Pilot, if there's any way to grab some of the coral rubble, we might be able to get a sense of what that is and even find one of these agglutinated foraminifera that are attached to it.
So grab some rubble too, watch it from the bottom, or try to.
Yeah.
Yeah, sure, thanks.
I would imagine that would have to be two separate things because we only need a tiny piece of the octocoral.
So it's pretty small. I'll put the arm next to it so you can see.
Okay.
Yeah.
Yeah, again,
right,
this is
3, 2, 1.
Would there be objection to getting a little, I don't want to say a scoop, but a scoop would give us also the sediment and the dead coral.
We could try
put
to
it
scoop
in the same bio box, potentially.
But with a current,
I guess so, yeah.
It might blow away. But I'd be
we
more
may
than
be
happy
blown
to
away
try
by
it.
the current, so perhaps we could do this at another location. We'll see if we have time.
Or if this works here, then we could get 2 different types of samples with one.
Yeah, it's great because we could put them in the same bio box since they obviously cohabitate.
Pilots are free.
All right, can we center?
Yeah, that's actually a lot smaller than I was thinking.
And zoom in slightly.
Once again.
So you're trying to take up a whole collection of like rubble and the coral?
I was just gonna still snip the coral.
Okay.
All right, watch it, Zed. Do you see the coral size now?
Yeah, that's a lot smaller. If you want to just grab that from the base and grab some of the coral rubble with it, that would be wonderful.
Copy that. We will attempt that, and then we'll probably just go port inner. That's good. So I'm just going to try to snip it, and then some of the rubble might get caught in there.
So I think they're saying just kind of close your jaws where you are.
I don't have it, but I will.
Take the whole
I'll
coral
log the
along
collection
with the rubble.
And it's been pointed out that this species is actually fairly common. So again, that makes us feel better about taking an entire small organism, and we can learn a lot more about the morphology by taking a whole sample as well. We don't do that often, especially with larger corals, but pen.
All right.
Unicella.
All right, Copilot, do you think you can open drawer here, or do I need to move? All right. Just hold for a second.
Maybe come down on minis. Yeah, that's pretty much most of the way.
We definitely got some of that coral rubble. So hopefully we'll find some agglutinated foraminifera that Chuck Messing has noted. And that is going.
So we are at 731 meters collecting this Unicella octocoral and a little bit of coral rubble, which is the habitat it's growing in.
I need your wrist a little bit.
Do you want to zoom on HD2?
No, that's all right.
If you bring your wrist up, you'll become more vertical. There you go.
And the big vase sponge you see off in the distance in the left-hand side looks like a rosellid vase sponge. Chris Kelly gave us that information. Big, beautiful sponge. It looks like a bamboo coral on the right center.
You can knock that little piece in. It's right on the edge of the drawer. You don't have to. I'm giving you a chance to do a little practice. Nicely done.
All right. Copilot, you can close that.
All right. Well done, Sean.
Very nicely done,
Sample
pilot.
is marked.
We were watching on the other camera while the pilot was very gently releasing the specimen, and then there was a piece of rubble on the side of the bio box and ever so slightly tapped into the box.
That's all right.
Hard to believe that we are more than 700 meters above, and the pilot is manipulating from the surface. All those gaming skills have come into play. So we have our first collection of the day. And we got the bonus of some of the coral rubble and maybe even some sediment attached, although I don't